Hello. Can we please get settled into our chairs so that we can start the session? Are there people outside who want to come in? Okay. It's on. Sure, absolutely.
Just a couple of announcements to reiterate. This lectures are beamed to LH1. So those of you who are in LH1 know about it, but if it gets crowded at the back, you can tell others to go there. There's Wi-Fi access in the hallway, but more important, there are also restrooms on the other side, not just here. So in case it gets crowded here, you can also go down. One final point, when coming down, please watch your step. Once the chairs are in here permanently, it'll be easier, but otherwise the edges are red on red and you might trip on them. So be careful.
Okay, I think we can start the session. And I've taken permission from the organizers to take a couple of minutes before we actually go into the talks. First of all, I want to thank everybody for being here. And I particularly want to thank Obed, Vijay, Krishnan, and others for giving me this opportunity to come here. It's a profound privilege to be at Obed's 80th birthday celebration. I also want to thank the organizers for giving me the honor of writing a message on the excitement that we had during the early days of neurogenetics at TIFR. And what you would see if you read it from that is that the excitement was so much that it's beyond expression. It was really a very exciting time. Whatever I learned in science, and I really mean whatever, how to do, what to look for, how to look at the larger picture while still looking at the small pieces on how to put together, I owe it to Obed. It's something which cannot be described. It's something which you learn only by being in the vicinity of somebody like him, not from the test tubes in the lab. Of course, if you talk to my family, they would think that I'm a gone case because when I joined Obed in 1973, I would go home and they'll ask me, what do you do? I would say, I kill flies. And they'll say, well, we all do that. What do you do specifically? And I'll say, I kill flies. And now when I go home, they know I'm a gone case, that I'm still killing flies after 40 years of being with Obed. One thing I want to mention about Obed is that he has a very special quality. He takes a challenge and meets it exceptionally well. And then he doesn't rest on his laurels. He seeks even more difficult challenges, whether it's in terms of setting up an institute or making seminal contributions. And here I want to add something which Obed would relate to. Ali Sardar Jafri must have Obed like people in mind when he said, "Thank फिर जज्बाए बेताब की आलम पे कमंद एक ख्वाब और भी ए हिम्मत दुश्वार पसंद." Translated, it means cast again the lasso of your restless passions over the world. One dream more, O lover of intractable challenges. When I thought of coming here, I initially had an illusion that I would say a few words about his achievement, but very soon realized that he cannot be encapsulated in a list because most of his contributions go beyond the bricks of an institute or papers he published, they're mostly intangible. So I would not even go there. What I would do instead is give him my profound thanks, because whatever many of us are, whether in science or in life, is what we have learned from him. And you don't teach these things in a lecture, in a PowerPoint slide, or anything of that nature. You teach it by living a life, for being a person that you are, and for a life which is really being lived that well. I hope all of you join me in saying congratulations to him on his 80th birthday, but not only to him, but basically all of us who have been touched by him, and to the world of science in general. So having stolen a couple of minutes from the speakers, I would come back to the earth and start the session. What you would notice from the sessions which I did myself, that most of the talks, not only in this session, but otherwise also, are on creatures like flies, rats, crabs, and so on. And I also think that very few people work on human neuroscience. And I think I know the reason for that. Because as you climb the evolutionary tree from flies, worms, zebrafish, crabs, rats, and so on, before you reach humans, you reach an intermediate species that's called the politicians. And when you're looking at neuroscience of something, you hit a void. And if you don't believe me, all that you have to do is look at the primary contest that's going on in the presidential election in the USA at this time. So with that, I will request the fly person, Subhabrata, to start his lecture. Thank you very much for your
Am I? It's probably not on. Let's see. Should we minute up, right?
Yeah, it's on.
Ah, yes, it's working now. I think.
25-minute lecture, and the bell would ring at 20 minutes. And what I would like to warn you is that there is a new mechanism for this session.
We get killed.
Drops at 20.
Well, I'll come down to the ground. Welcome back from tea. I would second Satpal's sentiments absolutely and say that it's a great privilege and honor to be here. Also, I would add that coming to NCBS is a strange mix of feeling relaxed and stimulated at the same time, kind of a strange cocktail of caffeine and alcohol. But let's go right ahead and get into the talk. So what I'm going to try and do is talk science, of course, but as Vijay mentioned, sort of go through my own experiences and my own intersections with Ubaid from the time that I was a grad student. So the way it all started was when I came for the graduate student interview, and NCBS at that time held joint interviews with the Bombay section, and I had only opted for the Bombay group. And I made two major gaffes, I think, in spite of which I got selected, which was interesting. The first one was during the interview, Veronica said, "So what kind of genetics have you done?" And I said, "None." So she said, "Well, what about fly genetics?" I said, "Still none." And Krishnan was there, and he kind of tried to rescue me by talking about yellow tigers and white tigers and so on. So I felt so relieved. And I felt so relieved and indebted that I plagued him by joining his lab after that. But the second major goof was Vijay saying, "You've realized you've only opted for Bombay and that cuts your choices by half." And I said, "Well, I don't particularly like South Indian food." And so it's kind of ironic that in Atlanta now I take special pains to go and eat South Indian food. But as it happens, I got selected, joined the Bombay branch, and got into Krishnan's lab. And very soon, as Chungfang was also talking about, some of the things that you sort of learn very quickly, and one of the reasons why I really joined that lab is you realize that conditional paralytics, these TS mutants, are probably one of the best things to have happened to the world of synaptic physiology. And it's a shame that in the fly world, at least these days, people don't do more EMS mutagenesis, because it's very hard to actually get this kind of mutants by looking at deletions and so on. But the idea was that you could really just observe. It was such a simple thing. You take one of these flies, para, for example, you heat them up, and these flies just collapsed. It was a simple demonstration, but it brought home the point that here's a mutant. This is a single gene. This is a change in one base pair that gave you such a profound, easy to observe, and really rather dramatic phenotype. So that's what basically hooked me into Krishnan's work. And at that time, most of the work at that time was going on with Shibiri, and comatose was kind of lurking in the background. And I don't know what the history was. I don't know whether people were not trying to look at comatose, but whatever it is, Krishnan convinced me that comatose is what I should be looking at. So comatose is what I looked at. And I'll sort of show the same manuscript that Chungfang mentioned, which is one of those classic manuscripts that everyone who begins the field kind of goes back to. Have you looked at Upinder and Seymour's manuscript? So this is one of those manuscripts. And what's shown here are findings with comatose. Chungfang, I think, talked about Shibiri a little bit. What Upinder had done is to show that just like the behavioral paralysis in comatose is fairly slow as compared to Shibiri or para, if you stuck an electrode and you raise the temperature of these animals were slow to lose their action potentials, and they were also slow to recover them. And that's really all that we knew about the physiology. Comatose flies paralyzed. This was their electrophysiological defect, period. At the time that I joined the lab, people had actually figured out what comatose is. And as was mentioned previously, comatose encodes the fly homologue of this protein called NSF. For a while, I took great pride in being able to say N-ethylmaldehyde sensitive fusion factor. That was my main key pitch. But comatose codes for NSF. Turns out that there are two NSFs in the fly, and to basically make a long story short, these two NSF homologues are expressed differentially. So NSF1, which is comatose, is expressed in the nervous system, NSF2 mostly in the mesoderm. So you lose NSF1, you've basically lost synaptic vesicle fusion in neurons, and that's how the fly paralyzes. So this is where we were. And the main question was still, although a lot of work had been done in yeast and looking at fusion from the trans Golgi, for example, we still didn't quite know what NSF did in neurons. One of the ideas was that this was a classic fusogen. So this was a protein that really was implementing. It was an ATPase, so this was the power for the vesicles to fuse at the presynaptic membrane. But that was far from clear. So that was kind of my mandate to try and at least add a little bit to the story of what does NSF do. And during one of those times, I had sort of a loosely defined committee. There was Krishnan and Veronica and whoever else was present. And on one such committee meetings, Vijay had just flown into Bombay. So this was at 9 p.m. or something. So I've been waiting the whole day. I know that Vijay's going to be there and we're going to have this committee meeting. So I'm waiting. And he walks in at 9 and he says, okay, fine, committee meeting. And so we talk about all of this stuff, and Vijay says, well, you really need to do some EFIS. And Krishnan says, you really need to do some EFIS. Veronica says, absolutely, you need to do some EFIS. So I said, fine, I need to do some EFIS. And the second thing they said before they said, okay, fine, we're off now, was to say, you should go talk to Upinder about it. That was a bit of a problem. Why was this a problem? This was a problem because if you were a graduate student at that time, Upinder had an aura. Upinder still has an aura. Upinder has always had an aura. But this was a gigantic aura. I mean, it's not just girl students who found him good looking, guys found him good looking too, wanted to be like him. We all failed miserably, but that's another story for another day. But the thing is, he had this huge aura. So I said, well, this is going to be tricky. I've never really spoken to him. We've sort of seen him from afar. And now I have to screw up the courage to go and talk to him about this stuff. And I spent a lot of time thinking about this. There were two ways of doing this. One was the normal way. If you had enough spunk, you would just go to his office, knock on his door, and say, can I please talk to you about this stuff? I didn't do that, too scared to do that. So I basically said, okay, let's try and first of all make sure that he's in. He was traveling a lot between NCBS and Bombay those days. And let's first make sure when he's in. So all these excuses you build up in your own mind. And one of the ways in which we knew he was in was by looking at his red car. So for almost a month, I would walk in and say, okay, the car's there. I just need to go and talk to him today. And an additional paradox, in addition to what Chung Fang mentioned, was we could never figure out how Ved got in and out of the tiny little car. Anyway, we'll ask him someday. But for a month, I looked at the car, I would walk in, I said, today is the day after lunch. Okay, fine, after tea. All right, in the evening if he's there, really, really. And that didn't happen for a while. One day, Shobna met me and said, you know, at this rate, your PhD isn't going to get done. So you really must go talk to him. Fortunately, I was saved from that because one day in one of my annual work seminars, I talked about comatose. Ved was right in the front, and he comes up and says, okay, let's talk. So we spoke for 2 hours after that. And ever since that time, we had many discussions, I mean, these were in his office, these were in the lab, of course, these were at the canteen. And there were some days when I would be walking in and he would be driving off in his car, he would actually stop and say, so what happened to that recording? So, you know, so many discussions later, he took me on this sort of a grand tour of doing electrophysiology. I don't think we had an E-phase rig to do neuromuscular recordings at that time. So we opened this door, which we'd seen on the top floor and which we'd never gone in. So we had no idea what was behind the door. So he took out his keys and opened the door, and there's this, you know, this treasure trove of things. And the task was to build a rig from all of that. And so that's the rig we built. It's a very simple, very effective, small but mighty rig. And one of the interesting things was that I have never seen, for those of you who do EFIS and who use suction electrodes, I have never seen a more carefully crafted bronze suction electrode. I have never seen one after this, and I need to go back and find it so I can keep it. But this was the rig, and basically one of the ideas was to look at more mutants of comatose and try and understand what the synaptic defect might be. Shown here is also the first trace. This was probably one of the most happiest days of my life. You do something simple, doesn't work for a long time, and it works. Absolute eureka moment. I have this Polaroid photograph even now from which this is a scan. Anyway, so this started working, and one of the other things I want to sort of stress is that, you know, in addition to being really taught by him about how to do this, the sort of the nuts and bolts of the whole process, those discussions were amazing because you sort of let go of the small worries. You could let go of the fact that your stock wasn't growing. You could really sort of expand your thinking and understand, well, if I get this result, what does it actually mean? What piece of puzzle does it solve? How does it fit into other stuff? These were the kinds of realms that he would just wander off, and you would just sort of sit there and go on this ride with him. So anyway, so we started looking at these recordings, and one of the ideas, as I mentioned, was to try and figure out what NSF actually does. And of course, these alleles that were isolated earlier on were going to be very useful. There were some additional alleles that Krishnan had isolated, and we were going to look at those as well. And in the midst of all that, this strange thing called the hutment came up. Now, for the life of me, I don't know why that place was called the hutment, but it was this sort of ante-room kind of thing, which was right across from the TIFR campus. It was fine. That was not the problem. The problem was that at some point in the middle of, I just gotten the damn thing to work. But then we had to move just that rig to the hutment, which sounds very simple, but it wasn't, because it was right in the middle of Bombay monsoon. So here we are. This was a bright sunny day. It usually wasn't like this during the monsoon. Here we are trying to drag this rig with all its contraptions all the way across this gale speed winds blowing rain, and with our umbrellas and so on. It's just an absolutely monstrous sight. Anyway, so we dragged the rig all the way down to the hutment, and that's where the experiments finally started happening. So one of the first things that we kind of did was very simple, and basically showed that NSF at that time was thought to be really the fusogen, as I mentioned. So this was a protein that was thought to cause the fusion of the presynaptic vesicle with the presynaptic membrane, and then that's it. That's what it did. And so if you lose NSF, you don't have vesicle fusion. That's all it did. And we had this strange result using, mind you, the same same alleles and paralytics that were isolated so many years ago, we basically showed something very curious. That is, first of all, in comatose animals, when you raise them to restrictive temperatures, these animals started firing wildly before anything stopped. So that was kind of strange. I mean, if this is a fusogen, why should these animals keep firing? That's one point. You would just assume that it would behave almost like para. There would be no action potential. This thing should just halt. That didn't happen. So first of all, comatose started firing before it collapsed. And secondly, you could do a strange, weird trick that you could only have conceived of in Krishnan's lab, which is if you combined two different mutations, temperature-sensitive alleles of comatose and para. Now, these things have different restrictive temperatures, para being the sodium channel. These animals stop firing action potentials at a lower temperature. So what you could do is you could shut down action potentials before you hit the temperature at which comatose would paralyze. And if you did that and brought it back down, then these guys at normal permissive temperatures would fire a random burst of firing that looked just like comatose and then paralyze. So then we did some more behavioral experiments, and basically we figured out that it cannot really be just the fusogen, if not the fusogen at all. It could not be just the fusogen, because what it seemed to be doing was allow quite a few rounds of action potential propagation, vesicle fusion, and synaptic transmission before things collapsed. And as it turns out, many experiments later from various labs and various systems, that's what's generally now believed to be true. NSF is not just the fusogen. In fact, it is involved in snare, well, to get into technicalities, it's involved in P-snare, B-snare combinations as well as splitting them apart. So it actually plays a role at the time of vesicle fusion and also at the time of vesicle retrieval. So we were able to significantly modify this model where it's actually shown as a fusogen to say that NSF has multiple roles all along this cycle of synaptic vesicle fusion and retrieval. And these are really very simple experiments. These traces, again, were all Polaroid traces. We had the little screen and we had our Polaroid, and these were all scans from that Polaroid. And then, you know, this burst of firing was then, I continued to use this when I moved to Mani's lab, and even in my lab after that. We've sort of continued to use this idea or this phenomenon of comatose animals firing more to various ends. First of all, we showed, for example, that you know, you could actually see this firing. So this is comatose. Comatose keeps firing at this temperature, and we could then combine it with other interesting mutations that cause more calcium release and so on, to show that, you know, it was interesting. It just wasn't firing. It turns out that these animals, because of their elevated firing, show reasonably strong chronic plasticity. So these animals end up getting synapses that are larger and stronger. So they release more neurotransmitter. That's shown here, for example. They actually release more neurotransmitter in a very stable way. And you know that this is because of activity, because you can suppress activity in the background of these mutations and show that this goes back. The other thing we sort of confirmed, so, you know, so are these animals hyperexcitable? Do they move around more? And Chung-Kwang was talking about EAG shaker and so on, and it turns out that these animals actually also move about a fair degree more than control. So shown here, for example, are basically activity profiles in one 24-hour period in a light-dark cycle, and the light gray bars are control animals, and the dark gray bars are basically comatose animals, and the amplitude of activity per, I think in this case it's about half an hour or so, is shown by the amplitude of the bar. So if you combine this over a period of a day, these animals have almost twice as much activity. So we've been able to exploit comatose not just for looking at synaptic transmission, but being able to use to leverage this phenotype of their firing a lot more as a means of inducing long-term plasticity. So once I sort of did all of this, so my movement through all of this was looking at synaptic physiology, trying to understand the role of NSF, going into plasticity and so on. And I kind of realized that I never really have left motor behaviors somehow. I mean, I was trained more as a molecular biologist, whatever that means. But no matter where I go, this business of motor neuron firing and motor behavior in some way just keeps coming back. First of all, because it's absolutely enchanting. Second of all, I mean, with these TS mutants, as I mentioned, these are just clear-cut, absolutely gorgeous phenotypes that you can study very easily, absolutely black or white. And in the little story that I'm going to mention now in my lab, we've again found ourselves looking front and center at at motor behavior of an interesting variety. So, Satpal was talking about us not studying humans. We're studying flies and worms and rats and mice and so on. This disease comes from humans. Turns out that there is this disorder, if you can call it that, called Restless Leg Syndrome. None of you have it, trust me. If you did, you would not be able to sit and listen. And the symptomatic diagnosis of Restless Leg Syndrome essentially is that these patients will not be able to sit still for any length of time. It gets much worse at rest. It gets phenomenally worse at night. And one of the predominant outcomes of Restless Leg Syndrome is very disturbed sleep. So what's called sleep consolidation is very badly affected. All of you know, I think, from any number of experiments you've done on yourselves, that if you don't get a good night's sleep, in terms of the quality of sleep or the architecture of sleep, next day you're a mess. And so it's not just the number of hours you get sleep, it really matters what kind of sleep you get. And Restless Leg Syndrome patients essentially have had a whole lifetime of having very, very bad, very disturbed sleep. All right. That's really all that's known about Restless Leg Syndrome. It affects about 5 to 10% of the population to varying degrees, so it's fairly widespread. It's not lethal or anything, so it won't kill you, but it absolutely destroys your quality of life. One of the ideas of trying to figure out, so what is Restless Leg Syndrome? Is it a motor behavior that gives you sleep phenotypes? Is it essentially a sensory motor disorder that disrupts sleep? Is it a sleep disorder that keeps you awake? Is it something like pain where the gating from the peripheral nerves is altered? And there are people who believe in all of these models. So we don't really understand what Restless Leg Syndrome is. One of the ideas was gleaned from the observation that RLS tends to run in families. So there could be a genetic component. Long story short, there were actually 5 genome-wide association studies done, 4 different loci identified as risk alleles, and we started working with one of them. It was a huge stretch at that point to try and even ask whether something like Restless Leg Syndrome can be mirrored in flies. But the basic point was that if you can measure the outputs, if you can measure the key symptoms of RLS, then you might be able to model this in flies. All right, so the gene that we started working with doesn't mean anything to anyone, didn't mean anything to me. It's a gene called BTBD9. It really doesn't mean much, apart from the fact that it has a domain called the BTB domain, which about 400 proteins have them. So it didn't give us much of a clue. The fly gene is called CG1826, which basically means that it was a hypothetical gene. Nobody had done anything with it. Okay, so one of the first things we decided to do, use your usual strategies, knock the gene out, try and see what the fly does. And in so doing, we were focused on sleep at that point because we knew that any model, if it had to recapitulate RLS, should have disturbed sleep. Either that is a primary phenotype or that is an outcome of defects in motor control or motor behavior. And sleep at that time had been fairly well studied in flies. Systems to measure sleep, what constitutes sleep in flies, what are the different things that affect sleep, things like dopamine had been worked out. So we had a reasonably good idea of what to look when we measured sleep in these mutant flies. Now, knocking out this gene doesn't kill the fly, it's still completely viable, which is good. If it's dead, you won't really be able to do an experiment with it. So here's sort of a summary of different phenotypes that we found in our so-called RLS mutant flies, and it was absolutely striking. The postdoc who was doing this work basically had a contract. I said, you can work on plasticity for 6 months and you can work on this thing for 6 months. I knew this wasn't going to work, so after 6 months she would be working on plasticity, great. That's not true, and we actually have moved away significantly from plasticity, and we work on this now, and that's because of these data. So, for example, shown here are 2 different mutant alleles. We call these flies Wanderlust, and you'll see why. So Wanderlust 1 and 2 basically have the same total sleep time. But look what happens at night. If you look at the number of times they fall asleep and wake up, so each time they fall asleep is a bout of sleep. So the number of bouts that they have. If you sleep totally for a long period of time, that's just one bout. If it gets disturbed, you'll have multiple bouts. So what happens is, in the 2 alleles, the night bout number goes up. What that means reciprocally is that each bout is very short. And so the bout length, the average bout length in both the alleles goes down. And what you can do is you can supply the wild-type protein specifically in the nervous system, and these phenotypes go back to being normal. So that tells you this was actually a fairly big surprise. I mean, what are the chances that you're going to go from a GWAS to getting a phenotype like this that's really strong and consistent with RLS? So then the final thing shown here is called wake after sleep onset, which basically means what length of time are you awake after the first time you fall asleep on any given night. And that's also significantly increased in the two mutants, and it goes back to control with the rescue. You might be able to appreciate these phenotypes a lot more from looking at these traces. These are what are called hypnograms. So these basically plot the sleep-wake transition in one day. Okay, so here are two wild-type flies. So they sort of fall asleep, wake up, fall asleep, wake up, fall asleep, wake up, and at night they basically sleep soundly. And here are two traces from two individual wanderlust mutants. These guys can never really fall asleep soundly at all. So they have classic sleep fragmentation. This set of graphs basically shows you that you can induce these phenotypes by knocking out this protein selectively in the dopaminergic system. Absolute surprise. Because guess what the frontline strategy for management in RLS patients is? Dopamine agonists. Nobody had ever shown any kind of connection, any kind of mechanism by which this gene can cause sleep phenotypes. And here we have selective knockdown in the dopaminergic system giving you the exact same phenotypes. But here's the real clincher. When we looked at motor behavior, so RLS, so you go to meetings and people say, well, do flies have restless legs? They say, well, you have 6 legs, we don't really know, we can't really tell. But what happens in these flies is that they basically have increased motor activity. And we've measured this in multiple ways. You can just track individual flies, and you can look and see that the two mutants walk a lot more than controls. We can do more sophisticated analysis as flies walk back and forth between two black bars and show through more in-depth analysis that what happens is that they don't walk any faster. They walk at the exact same speed. The only thing that changes is the number of pauses they take. They pause less. They continue to walk more. This is absolutely uncannily similar to what you might call a restless phenotype, right? So I'm going to end this story by just giving you a flavor for the story. We've done a lot more molecular work, and I'll be happy to talk about this afterwards. But basically, this gene controls ubiquitination, and we think that has something to do with the way dopaminergic neurons function. We're going to sort of stop and sort of recap and go back to Ubaid's contribution. I've already mentioned all of these things. And I would just like to reiterate that there were two main things. First of all, learning electrophysiology from him really made me overcome the fear of electrophysiology. I had never done that before. And he just took me through very simple steps. He just made it sound very simple. He said, you know, you do this, you do this, you do this. And we did it. It worked. And after that, you get the confidence to try out more and more complicated things. So that was a major threshold that he helped me cross. The other one, as I said, is, you know, talking to him, it really sort of expanded your mind, and it made you think much bigger than your tiny contribution. And I've sort of tried to always keep that in mind. And finally, of course, I'm still trying to be as good looking as him. That's not working very well, so we'll see about that. But I'll end there, take any questions, and I'll thank people at Emory. These are people in my lab. We have very productive collaborations at Emory and continue to collaborate with a lot of people, all of who bring in great ideas, great challenges, and just stop us from resting on any so-called laurels that we might have had. Thank you for your patience. Thanks.
Thank you very much, and we have questions. Dr. Rao?
Is there any possibility of the whole pathway by doing something that might be, you know, akin to an RNAi screen in whole flies?
Whole pathway for?
So you have a BTB domain protein.
Right.
It's presumably part of a Cullen complex of some sort.
Yes.
That would have a substrate and something that controls.
Absolutely. Yeah, exactly. So I think the business end of the whole system is going to be that. And what we're doing is a SILAC-based screen to look for binding partners of BTBT9, and then followed up by phenotypic analysis of BTBT9. Absolutely, yeah.
So Sanyal, well, if you knock this down in dopaminergic neurons, you get the phenotype. But can you get it by knocking it down in other neurons as well?
You can get it by knocking it down in all neurons. You cannot get it by knocking it down in motor neurons, and you cannot get it by knocking down in glia. So we think by default it's dopaminergic neurons. These are all THGAL4 experiments. We've used the different classes of HLs. Those experiments are not complete, but I think we have identified two of the very many clusters of dopaminergic neurons where you absolutely need to knock it down. It's a little more complicated because rescue doesn't quite work that way. So you need to really rescue in all dopaminergic neurons.
Can I ask something? Yeah. You observed that walking behavior in adult flies, like which increases when they have
Right.
mutation. Is it also prevalent in the larval stages?
We've not seen anything. That's point one. Point two is that BTBD9 actually has very low expression in larval stages. It really seems to be an adult expressing gene. Nobody, to my knowledge, has looked at larval sleep. The only thing that comes close to sort of larval stage sleep is in C. elegans, is a stage called lethargus. But there is still debate as to whether that's really sleep. That's another story. But short answer, no. I don't think BTBD9 is expressed in larval stages, so we haven't really looked. We haven't seen anything absolutely obvious.
Any particular reason, like why that gene is expressed only in the adult stages? Like it has to do something with the...
Well, as was mentioned, I think it has to do with the ubiquitination pathway, and unless we figure out what the targets of this protein, this BTBD9, it's a substrate adapter for TALN3. We know that, and so what it does is it probably binds to very specific proteins in specific cell types, we think, and controls their ubiquitination. So it's a long road ahead. We need to figure out what the substrates are. We need to figure out what the ubiquitination of those substrates actually does. We do know that in the mutants, we actually have 50% reduction in dopamine. So this dopamine connection is actually pretty strong.
Any other questions? Okay, we'll move to the next talk. And while Gaiti is coming on stage, I want to vouch for what Eve Harder said and counter what you just heard. I have been having a lot of restless legs sitting here to come to the stage and talk about what Obaid taught and what he means to all of us. As I hear more and more about what is being said here, I really want to come up. But it's the inhibitory circuits that she said that they modulate all our behavior. And thanks for that, I'm not interrupting more. Gaiti would be speaking on the genetics of calcium signaling in flies. Thank you.
Thank you, Satpal. So before my slide comes up, I should really say that I think the title was chosen for me by Vijay. What? You would never do that. So I changed the title, inevitably. But I should say why I think how we came about. So I came into flies thanks to Obaid really many years ago. I was a molecular biologist. I did my PhD as a molecular biologist. I wanted to use molecular biology to study, genetics and molecular genetics. And Obaid at TIFR offered the possibility of actually being able to do that back in India at that early stage. This was in 1980s, sometime in early 1980s, 1983, I think, to be precise. And I already knew about this because I had visited TIFR as a summer student a couple of times, spent time working with flies and, watching Veronica doing interesting experiments, behavior experiments. And it was really, it was so exciting to think about the fact that one could actually look at molecular biology and then go to behavior. And that has really been the driving force of my lab in all these years. We've looked at many different things. And I thought, perhaps I should talk about the olfactory work, which we still do to some extent in the lab, mostly in collaboration with Veronica. And now that she's no longer there, it's sort of gone a little downhill, I should say. But we have primarily in the lab, I worked on flight more than olfaction. And I sort of, dithered, should I talk on olfaction, should I talk on flight? And then I decided, okay, never mind, let's talk on something completely different, which we've been doing more recently. And, let's get some, feedback from other people. Hopefully what I'll be able to do is to explain how, looking at molecules is really, and going to behavior, how looking at the whole picture is what one has really learned to do over the years, and which is essentially what, was the idea, I think, of looking at molecular biology and olfaction originally by talking to Ubaid. And so I'll go into the story right now. So what I'll talk about, rather than either flight or olfaction, is this new story, which is on looking at IP3-dependent mechanisms that coordinate appetite and obesity in flies. Just to give a little introduction, so what we work on is intracellular calcium signaling primarily. Cells can get calcium from two sources, either from extracellular sources, which come in through channels and pumps, or by stimulating a receptor and release from intracellular stores. And one of the main things that our lab has been interested in is trying to understand cellular functions that are coordinated by intracellular calcium release, particularly in neurons. That's been the interest. Why is this important? Well, one of the reasons we think this is really important and should be looked at carefully is that all molecules that are involved in intracellular calcium signaling actually evolved at the time when multicellularity came into existence in evolution, which is at the stage when choanoflagellates develop, and then you see them at subsequent genera. You do not see them in yeast, and you do not see them in completely unicellular organisms. And so this, we think, is perhaps a way by which this kind of coordination can take place in an organism, that using intracellular calcium signaling is probably one of the means by which a multicellular organism actually coordinates all these multiple kinds of activities to produce behavior and existence. The molecules that I'll talk about more today is one is the IP3 receptor, which releases calcium from intracellular stores. And I won't really talk too much about this today, but we also work with STIM, which is the intracellular calcium sensor, and ORAI, which is present on the plasma membrane and is the calcium channel, both of which were discovered in screens using RNAi in flies as one of the ways of screening for these new genes. And one of the people who discovered it, Anjana, is in the audience, and hopefully we'll hear more about it from her in her talk tomorrow. So the way that the IP3 receptor gets activated, what's known is that there are a large number of molecules that can generate IP3 in the cell. And this is both in neurons and in other kinds of cells this can happen. And I guess the question really has been is to try and understand in what context this signaling is important in terms of behavior. We do this in flies. We look in flies. And the way we did this originally was by generating mutants in the IP3 receptor. And these are some of the single point mutants that we found, that we isolated and then subsequently mapped in the IP3 receptor. And the way we got into looking at this aspect of it was that we found that there are growth defects in strong mutants for the IP3 receptor. And this is one of the, in the larvae, you can see that if wild-type larvae grow to this level, then the mutants essentially look very small, are quite sort of puny looking, and in terms of length are half the size. And then you can rescue them back by putting the IP3 receptor back in these flies, in these larvae. And interestingly, the rescue was primarily from this set of cells, which are the insulin-producing neurons, which are present, also called the medial neurosecretory cells, which are present in the brain of the fly. And these are the cells shown here. So IP3 receptor put back in these cells can rescue back the mutant to normal conditions. What we also found was that there's another phenotype, which is in viable mutants, IP3 receptor viable mutants, which are shown here. They have this wing phenotype and they can't fly. And actually, if you put the IP3 receptor back in the DILP neurons, even in the adults, in these mutants that are viable as adults, you can see that they can get rescued back. And in fact, if you look at their flight, that is also rescued. So mutants are 100% flight defective, and these are now rescued back to almost completely normal by putting the IP3 receptor back in DILP neurons. And so what we at that point, and this is all published work which we showed earlier, and what that suggested to us was that there is some way that by which intracellular calcium homeostasis in neurons, and particularly in the DILP neurons, is required for flight circuit development and possibly larval feeding and growth, and that there is a rescue that can take place by increasing perhaps function of the IP3 receptor such that both these aspects can get rescued. I won't talk about this today, but there's also, it seems that it can also be rescued from the aminergic neurons. And so what we really began to wonder was that there seemed to be a role for the IP3 receptor in insulin-producing cells in IP3 receptor mutants, and we wanted to know what is this, is there really a role and what is this role? And so what Mani in the lab did was he looked at some phenotypes which are sort of traditionally known for insulin mutants, insulin signaling mutants in Drosophila. So for one of them that he looked at was starvation resistance, and found that if you look at starvation resistance in the IP3 receptor mutants, they live for significantly longer under starvation than do the wild type flies. So they're living for nearly 72 hours longer, and then even the curve, the drop down is much slower, is considerably slower, and so they die considerably later as compared to wild type. And the question was, why does this happen? So we looked at their stores, at their triacylglyceride stores in their fat body. This is a wild type fat body, and you can see that these are the tag stores that are present in the fat body cells in the adults. And if you starve them, these tag stores drop. So this is essentially where the energy comes from during starvation, and they lose their tag stores. And essentially, but once these run out, the flies will die. In the IP3 receptor mutants, what we found was that there was a vast amount of tag stores, and essentially when you starved them, these didn't change very much. You could see that they looked more or less the same. This was quantified, and this is shown here. This is work now that was done in collaboration with Dominic Schwudke, who unfortunately is not here today. It would have been nice to have him here to sort of correct me in some of these things. But anyway, so Suman worked with me and with Dominic to do some of these experiments. And what he found was that the levels of tags were considerably higher in the IP3 receptor mutant that we were studying as compared to wild type. And that on starvation, they changed very, very slightly. And this is actually looking at just the biochemical levels of tags. And here you can see them actually visually by doing a thin layer chromatography. And this is the starvation in wild type, and you can see that in the mutants it's much higher. The tag levels are much higher here. This could again be rescued by putting the IP3 receptor back in the DILP cells, in the IPCs that I talked about, and you can see that these levels of on starvation, if you put back either the IP3 receptor or D-stim, which is the intracellular calcium signaling molecule, you can see in both cases that you rescue back the starvation defect, and in fact the profile also goes back. So this is the mutants, and these are the rescued, and they're just like wild type now. And this is just quantifying it again and showing that in fact with IP3 receptor, if you put it back, the levels drop both at feeding and under starvation. And this is the TLC showing the same phenotype. So what this leads to, these excess tag levels, what they end up doing is end up creating obese flies, because when you take their body weight, you can see that the mutants are way much more than the wild type, which is shown here. And essentially on starvation as well, this level drops only somewhat, significantly higher still than the wild type and the rescued conditions. So essentially we have ended up with an IP3 receptor mutant, we have ended up creating a fly which is obese on normal food. When we feed it normal food, it ends up being obese, and it has a huge amount of tag deposits. So we wondered whether this was possibly to do with the fact that insulin signaling was affected in these flies. Again, that's what we had originally set out to do. And so this is a cartoon of the IPCs showing that they would release insulin from here, and you would get, as a consequence of the insulin release, you would get this tags would be utilized, and so this level would drop. And this should essentially happen under starvation. And the question was whether the presence of the IP3 receptor in these cells, is that what is causing the prevention of this insulin release? So the way we thought of looking at this was by essentially checking for by raising essentially intracellular calcium signaling should raise the levels of insulin signaling in these flies. So what we did was we said, let's see whether this is really the case. And what we put back, just the insulin, the ILP transgene, that is the insulin peptide, which looks most like the mammalian insulin, which is ILP2. We put that back in the IP3 receptor mutants in only the insulin producing cells, and then look to see what happens to the fat body. And what you can see here again is that they do in fact rescue the phenotype, very similar to what we saw in the IP3 receptor, at least in terms of the starvation phenotype. Though if you see here, this level still looks a little higher than what it was, and this level is considerably higher than what it is in the wild type. I haven't got a comparison here, but if you look here, you can see that the levels of the tags actually still remain high in the mutants, even though they utilize them better. And so the starvation profile came back to normal, but what we did find was that the levels of tags were still somewhat higher than they should have been. And this suggested to us, and correspondingly what we find is that the body weight is still higher. So the DILT2 rescued flies, their body weight is still higher as compared to wild type. So this is the body weight of the mutant, this is the body weight of the DILT2 rescued fly, and this is still higher than what you would expect in the wild type at this condition. So essentially, if the IP3 receptor mutants were starvation resistant, they stored tags and their body weight was elevated. The IP3 receptor, all three things were rescued, but with the elevated insulin signaling, this was not the case. And what we found was that, in fact, the tags were still elevated and the body weight was still elevated. And so we wondered what was going on here. There was obviously not just insulin signaling which was altered, something else must be altered. And so at that point, what Mani thought was that perhaps it's the food ingestion which is changing as well. It's known that these insulin-producing cells, the medial secretory neurons, they produce other peptides as well, something that drosulfacanin is one of them, and that these are involved in actually regulating the level of food intake. So what Mani did then was to actually look at this part of the pathway, which is to see whether ingestion of food was altered in IP3 receptor mutants. And he did this by doing a classical feeding assay, which is something that a taste assay, which was done earlier, something similar to a taste assay, but was done a little bit differently, in which that you starved the flies for a while and then put them onto food with colored dye in it, and looked at the mutants and found that in the wild type, this is what the level of ingestion was, whereas in the mutants, you could see that the abdomens were considerably more red and they had a lot more red food in them. This is after ingesting food after 12 hours of starvation. And correspondingly, the midguts, if you stained them for oil red, which is for the fat, you could see that the midguts were stained with a lot more of tags than they were in the mutants than they were in the wild type. And again, this could be rescued by putting the IP3 receptor back in the IPCs. So this was quantified, and you can see here that the red dye is considerably higher in these abdomens than it is in the mutants than it is in the wild type. And again, the ILP rescue does not rescue this phenotype. So essentially telling us that the ILPs can rescue the utilization of tags, but the ILPs are unable to rescue the ingestion of food. That is, the IP3 receptor mutants are eating more for some reason and are essentially generating, keeping a lot more of tags in their fat bodies. The ILP can use up this, cause the utilization of these tags, but the excess eating still continues in these mutants. And so the question was, what is the reason for this? We also looked at the lipase activity, because if these mutants are eating more, then there has to be some way by which they take this food and actually generate it into tags. And what we found was that they have higher levels of various lipases in the gut. So these are transcripts of various lipases, and actual lipase activity in the midgut also goes up. So there are corresponding changes in metabolism which allow for the larger, sort of distribution of tags into the fat body. So essentially what we ended up finding at this point was that there was a loss of appetite control and there was upregulation of gastric lipases in the IP3 receptor mutants, and we still didn't really know why this was happening. What was driving the mutants to eat more and to have elevated body weight? And so again, we sort of went with Dominic, and we sort of went and discussed with him and said, we should really look at these, the changes in these tags and see whether there's anything actually changing in terms of, to try and get to understand what is going on. And so with Dominic's help, Suman did this mass spec analysis of the abdominal tags. And, we really couldn't see anything different. So the abdominal tags between the IP3 receptor mutants and the wild type were really very similar. All we knew was that they were getting less. That they were higher level, but their profile didn't look very different. But then when Suman went and did this slightly more, analyzed them at a higher level and tried to see what were the fatty acids that constitute the triacylglycerides, and were they changing in any way, he could see this very small change here, which I've shown you here, which is between the wild type and the mutant, which is that these the tags which come from the fatty acids 18.0, 18.1, and 18.2, they show a slight difference in the IP3 receptor mutant. So on starvation, while these go up in the wild type, these seem to go down. And this was the only small change that we could actually see in the IP3 receptor mutants. And so we wondered whether this was really the meaning. But what was comforting was that in the case of the rescues, these went back. So this made us think that there must really be something behind this. But the question was, what is this? What was the, what was the change in the C18? So we looked then at, decided to change the diet of the IP3 receptor mutants and put them only on sucrose and now see what happens. Do they show the same kind of profiles that we've seen earlier? So on normal food, if you look at them, you can see that the IP3 receptor mutants last for longer. This is just the time at which they die. 50% of them die after starvation. So you can see that this time is much longer, which is what I had shown you earlier. On sucrose, we found this didn't happen. On sucrose, they die at the same time as the wild type. And so this is exactly here. And when you look at the abdominal tag levels, that's just measuring the levels of tags. Again, on normal food, they are much higher. This I already showed you. And on sucrose, they are higher when they are fed, but they drop when they are starved. So this told us that there was some limitation in the way that, if you give them only sucrose, they are unable to deal with some aspect of their nutrient synthesis, and there's a problem. And taking this together with what we had seen with the C18, the fact that the fatty acid C18 was reduced, what Mani did then was to put back various fatty acids into the sucrose diet and then see whether he could rescue back the viability of these animals. And when he did that, what he found was that essentially you could rescue back viability to some extent, at least a reasonable extent, by putting back stearic acid, which is C18. And so this is what you're seeing here now. These are the mutants themselves. If you just grow them from the larval stage onwards on sucrose, then they can barely pupate, and essentially you don't get any adults at all. This is just the control here. If you put back stearic acid into the medium, then you can see that they pupate significantly to, nearly more than half of them can pupate, and a significant number of them actually emerge as adults. So essentially, stearic acid could rescue back some level of viability to the IP3 receptor mutants, suggesting to us that they were actually stearic acid auxotrophs, apart from perhaps other phenotypes. So then what we decided to do was to look at other phospholipids, because stearic acid is known to go into the synthesis of many membrane lipids. And so this was analyzed by mass spec. And here you can see in Cantonese on normal food and on sucrose, you can see that they have, there's not much change in their phospholipid profile for the phosphatidylethanolamine and these various lipids, all of which have in them some amount of this fatty acid 18 shown here. But in the IP3 receptor mutants, what you can see is that on sucrose, these levels are lower. So the red bars show that, and the sucrose, these levels are lower for all of these different phospholipids that are shown here. And gratifyingly, when you give them C18, you find that these levels do indeed go up. So, and again, indicating that this is probably the reason why these are getting rescued. So finally, I think what with this mass spec analysis, what we were able to actually understand was that the loss of the IP3 receptor in the neurons leads to a change in the way the whole lipid metabolism takes place in the IP3 receptor mutants. So once the IP3 receptor function goes down in the brain, this seems to affect the synthesis of phospholipids, particularly this class of phospholipids which use C18. And as a result of this drop, presumably there is a feedback mechanism which then leads to greater food intake, so that on normal food at least, this greater food intake, you can actually compensate for these phospholipids, the loss of these phospholipids. But when you go onto a sucrose diet, then as a consequence of that, then this actually begins to show up. And this greater food intake, as a result of that, there is this, what we see, the obesity and the starvation resistance. It's a sort of side effect to what the actual genuine problem in these is. Obviously, we now need to begin to understand exactly how this is happening, what are the changes taking place here in terms of IP3 receptor function, how does it link to the synthesis of phospholipids. We need to understand that, and we're sort of getting into that now. So I'd just like to finish now by mentioning the people. So Mani is the person who did most of the work, or most of the behavioral and the genetics on this. Suman did all the mass spec that I talked about in combination with Dominic. As I said, I was hoping Dominic would be here to show himself, but he's not, and I don't have him on this slide. Some of the other people whose work will be up in the posters tomorrow, Sumita and Tarjini will talk about their work in the posters, and Trambak as well. That's right, 3 of them. So I'll stop here and take questions.
What's the biochemical connection between the IP3 receptor and lipid metabolism? Is that understood? Or
No, it's not. No. So I think, as far as I know, it's not understood at all.
This was
I
a
couldn't
big
find
surprise when you...
This was a big surprise. Yes, it was a big surprise.
Gaiti?
Yes.
Yeah.
Yes.
So you're talking about IPCs, and I'm just wondering how compartmentalized this story is. So how does it fit in relation to, or have you given any thought, you must have, to looking at the usual suspects of FOXO and JNK?
So we did all that, yes. I didn't go into the details of that. We looked at FOXO and JNK. So they all work in the same way that the ILP, when we raised ILP levels, if you change insulin signaling by using JNK dominant negative in the brain, in the ILPs, then you get sort of exactly the kind of phenotypes that you get with increasing insulin signaling. So essentially, utilization of tags goes up in the IP3 receptor mutants. If you change insulin signaling in the expect, if you raise it, it goes up in whichever way you like. Okay, so you can drive a dominant active PI3 kinase in the fat body, and you will utilize, the utilization will go up. So what it suggests is that it's a secondary way of changing the utilization of tags, but that's probably not the primary defect. We need to obviously look at the actual levels of insulin peptides in the hemolymph of IP3 receptor mutants to see if those are actually less than they would be in wild type. We don't know that yet. I mean, it's a hard experiment to do, and we're sort of gearing up for that, but haven't done it yet.
Thanks.
So to follow up on both those questions, this is not a cell-in disease.
No, I don't
So
think
what
so.
you do is change the calcium signaling in some population of neurons.
Correct.
That's then affecting somehow maybe hormone release or something.
That's what we think. Yes.
Metabolism in the whole animal?
In the whole animal, presumably in the whole animal. And I think what we hope to now do is to actually do subdomains, express back in subdomains, and then see whether, we can rescue or, bring down with RNAi and essentially generate the same defects.
Right.
But yes, exactly.
So that's
yes.
sort of the same question I asked Dr. Sanyal before.
Yeah.
Can you do RNAi screens on a genome-wide level in the whole organism, in the whole fly? Like you do it in C. elegans, for instance. You just
Right. No, you can knock it down in specific cell types. You can knock it down in specific cell types. You can knock down using RNAi in specific cell types and then see what the phenotypes are.
I see. And you can't do it by just feeding them the way you feed C. elegans.
No, not that I know of, at least.
If I may just say, I think there are, at the animal level, there are RNAi screens. Sorry? Intracellular. As far as I know, I may be mistaken, but I think a very large number of RNAi have been isolated for almost all, I think 70–80% of the genes. But we can, yeah.
They exist, and just asking if there's some high-throughput way to analyze all of them in an organism.
Utpal wants to say something. There is a question there.
Yeah.
Yes.
I was just wondering, how do you link these ILP-producing neurons to flight control? Are they part of the circuit, or are they?
So no, we don't think they're part of the circuit. Actually, we don't understand that, quite honestly. We do not understand exactly how. So all we can say is that insulin, so they don't only produce insulin. These ILP-producing cells produce many other neuropeptides. And what I think, in a hand-waving kind of a way, I can say is that there are presumably some neuromodulators, neuropeptides which are neuromodulators which are produced, which can essentially lead to producing a circuit which functions in when we rescue from the ILP neurons. Because what we've shown is that we can rescue flight both from the ILP neurons and from other neurons. and which do not overlap. So there have to be two ways by which we can rescue flight and IP3 receptor mutants, two different ways by which we can do it.
I think I'm getting a signal that the time is really over, so I apologize for not taking further questions. Thank you very much, Gaiti. And I think the next talk is by Upinder, and he would be talking on rats smell in my stereo. Upinder, thank you very much.
Okay, so many speakers have already eloquently spoken about how Obaid has influenced their scientific careers. So I'll give you my little anecdote. So some 35 years ago, when I was a teenager in JNU campus, my father said, okay, there's this very interesting scientist visiting his sister, Imrana, who is in the next quadrangle. And so he sent me off to talk to Obaid, who was visiting there. And at that time, I was deeply interested in physics, and biology was not on my horizon. And so anyway, I had a pleasant discussion with Obaid. And I'm sorry to say that following that, I went on to do physics. But in contrast to many of the speakers who have shown a very wide range of things that they've done after having been so deeply influenced by Obaid, I actually subsequently went on to work in olfaction and olfactory behavior, which, of course, I obviously took a wrong turning somewhere, since I work on rats rather than flies. But still, there's that interesting convergence of paths many, many years later. So I'm going to tell you about this, and the talk title is not my fault. This was given to me, but actually it's appropriate, and you'll get to see why it is appropriate. So this is a description of work that we've been doing for many years now, looking at odor-guided behavior. That is how animals follow things up to the source, odors to the source, and this has been done by Raghav, James, Adil, Manaswini, Partha, and Urvashi, and funding from all these good folks there. And with that under my belt, let me now actually put the way that I would have put the title. This is actually expressed by Partha some years ago. So he made this marvelous poster based on a topical movie of that time, Lord of the Rats, The Two Nostrils. The journey continues. So there's our hero, Raghav, and that's James. And you can see Raghav is being Lord of the Rats. I don't know if you can see all the rats dancing to his tune. So these are the heroes of the start of the story, and here are the heroes of the more recent parts. There's Partha himself, the author of that poster. Urvashi, whose work I will not be telling you much about, and Adil, who has unfortunately been truncated. Let me see if I can address the situation a bit here. No, that didn't work. Oh, that's even more exciting. Okay, they say I have to log back in. Okay. This is where I forget my password. Okay. This is great, but there's no menu bar. Oh, there we are. Okay. Okay, and that should hopefully work. Hello, anybody home? It is not coming. I'm sorry?
It is down on the bar below.
No, there's nothing else. This is where it is. We'll have another go at it. Ah, success. Okay. Sorry about that. All right. Now moving merrily on. Okay, so those are the people, and now do we have all of Adil? Yeah, we have all of Adil now. That's good. All right. So animals are very good at following odors to their source. Here's how moths do it. So if there's an odor source over there, a female moth, who the male moths are interested in, the kind of dispersion of odors is actually very complicated. You get these plumes which depend on the air currents, turbulence, obstacles, and so on. And so moths have this strategy of casting back and forth across the odor trail and are pretty successful in finding the source. Here are some other examples of other animals. So this is an experiment done by Noam Sobel. Here's how dogs track an odor trail, in this case a dead bird, dragged across a patch of turf. And again, it's zigzagging across it. And this creature is an undergraduate. And the undergraduate tracks a chocolate trail, again by zigzagging across the source. Similarly, John Hildebrand has shown how moths and, in fact, even waterborne tracking can uses the same approach. So one expects that when there is a good directional cue, that two nostrils, two sensors are better than one. And this is actually a pretty common sensory thing. That is, you see with two eyes, that gives you an extra dimension to your vision. And you hear with two ears, which allows you to actually locate objects very closely. So why not olfaction? So this is an overview then of the use of two sensors as being better than one in tracking odors. So let me just jump right in. So this is the study that Raghav did on this. And just to orient you in the movie, I'll show you in just a moment. There's rat. Here are two odor sources. They're actually completely symmetrical, just this angle of the camera. And there's an airstream coming from both of them simultaneously. And one of those airstreams is odorized. You have here, just for the purposes of a movie, two LEDs, one there indicating that that is the side of the odor, and one there indicating that that is the side of the odor. And without further ado, let me show you what the rat does. Okay, so this is what the rat does. It sticks its nose in through the hole. In this case, it actually got it wrong. And decides which side the odor is coming from, and then goes and licks that side in order to get its reward, which in this case is just water, because water doesn't smell much. And the rat performs this task at about 80%, 80-90%. And it looks, at least at face value, that the animal is doing some kind of very, very quick sampling and deciding based on that where the odor came from. Okay, well, that's good. That doesn't confirm that this is stereo. And the way you do that, of course, is you block one of the sensors. And so that's what Raghav did. So here is the behavior performance for different rats in different symbols. Well over 80%. Then he blocks one nostril. The performance drops quite significantly. Unblocks it. Performance bounces back. Blocks the other nostril. Performance drops again, and back again. And just to confirm that the animal is not just so miserable at having one of its nostrils blocked or completely incapable of detecting the odor at all, he also did a much simpler task for the animal, where the animal was just supposed to report the presence of an odor. And that's this. And you can see that the animal is perfectly happy doing this task, even when one of the nostrils or the other nostril is blocked. So the performance is almost 100% in this case. So you don't need to have both nostrils active in order to discriminate. You just need it for localizing it. And I should add that he went through a whole lot more controls and so on, which I will skip over in the interest of time, to, I think, help to pin down that both nostrils were necessary for this task. And then he went and recorded from the brains of these animals and showed that the odor response in the mitral cells was ipsilateral, you can see. So this is a dot raster plot. Each of these little crosses is an action potential. This is the time when the odor is presented, and you can see that the activity is much higher, also shown here in a histogram, which is the same data just summed up. This is the odor on the ipsilateral side, and that is the odor on the contralateral side, and you can see that there is no change during the odor presentation. No, there's no significant thing happening. Okay, so after we published this, so this is where the title of the talk comes from. This was the most interesting commentary we got from on the blog site that they had set up for the paper. So that's what I had rat smell in my stereo too. It was gross, man. I've always suspected Vijay here himself put that in there. Vijay, do you confess? No, you didn't do it. Oh, yeah. Okay. Well, with that staunch denial. Anyway, so this was the most interesting commentary we got on that. Okay, so just to pin this down further, Partha has been working since then. Again, it's a similar protocol, but now the task is much harder. And so the question we're after here is, are the rats performing a differential comparison between left and right? And this is not such a simple matter, because if you consider the fact that your sensitivity on either side may be different, and in fact in humans it does vary. Your nasal flow actually varies over time. So now if your sensitivity is changing from time to time, then a simple differential comparison would actually be a very tricky way to identify where the odor was coming from, because sometimes this side would be more sensitive than this side, and so you tend to think that, oh, the odor is coming from that side. Well, let's see what happens. So the way we went about this was to ask, can rats discriminate arbitrary concentration gradients when they are given in a random order? And so the protocol was quite similar to what you saw earlier. Why don't I show you what the rats do? Okay, so there's the rat, similar kind of protocol, except that now it's a 3-odor task. So here's one odor, there's another odor, there's another odor. The LEDs indicate which side the odor is coming from. Let's see if you can figure out what the rat has figured out. What is the nature of this task? Okay, so it's performing at 100%, I should point out. Okay, anyway, so as not to keep you in suspense, there are 3 odors. If this odor, indicated by these 2 LEDs, is on, then the rat always has to go there. If it's this odor, the rat always has to go there. And if it's that odor, it has to go whichever side the odor came from, right? And on top of this, there is a gradient change for this odor being given in. So what this does is it allows us to ensure in one set of trials that the animal's discrimination is fine, even as it is doing the odor gradient detection task. So it's sort of including control in the experiment itself. Okay, so it turns out that the performance is pretty high. If the odor is coming from the left, regardless of the gradient, it goes to the left, and vice versa. And of course, interestingly, and this sort of comes to the key point about sensitivity on either side, if the odor is identical on either side, if you give the same concentration on each side, so I should clarify, the first number indicates the concentration on the left, the second number indicates the concentration on the right, and you can see we've tried different combinations. And so when you give a 1:1 thing, so 1% on either side, then the rat is just confused, which is interesting because this is exactly symmetric, despite all possible changes in sensitivity on either side. And it turns out that the accuracy is well above chance. Not surprisingly, when the gradient is quite weak, the performance drops a little bit, but not a huge amount. It drops significantly, but it's still well above chance. And again, not surprisingly, as the task becomes a little bit harder with the smaller gradients, the time it takes also increases a bit. And of course, if you confuse the rat, then it takes longer. So Partha has also gone and done some physiology. This is intrinsic signal imaging on this. And this asks the question, is if you deliver a certain concentration on one side and dial up the concentration on the other side, does this affect the response to the ipsilateral side? Does it affect the response to the first side? And so what you have here is on this side, on the right-hand side, the concentration is always 2%. And the concentration on the left-hand side is going up and down, going from 0 to 2%. And the little black dots are indicative of responses, intrinsic signal responses. This is a picture of the olfactory bulb, a picture of the responses to the odor. And as you can see, as you increase the concentration on the left side, the number of little black dots increases. Small number there, larger there, larger there. But, and this turns out to be absolutely unambiguous from the statistical analysis, the response on this side is completely unaffected by what is happening on the contralateral side. And this happens for different odors, happens for different rats, for different concentration gradients. It's completely independent of the contralateral side. So to cut a very long story short, Partha did a whole lot of checks, further analyzing the possible changes in airflow and all of that. And quite remarkably, it turns out that the rat, at least, has got an almost perfectly symmetrical response, and that the airflows don't seem to change, and this is completely consistent over time. So in other words, the problem itself seems to have been solved by the rat by maintaining a pretty precise balance between left and right, so that a differential comparison works very successfully. Anyway, so just to summarize this part of it, rats do smell in stereo. They do a differential comparison. They sort of subtract one side from the other. They do this very rapidly, which has actually got some interesting implication, and that the left and right sensitivity remains in very fine balance. Okay, so let me now change gears and tell you a little bit about going a little bit more towards what I would like to think of as a more natural context, which is here we've been putting rats in a box and forcing them to sniff odors presented through a little hole. What happens if they can go and track down the odors? So this has been done by Urvashi, who's doing a very difficult experiment involving airborne odor tracking. And I won't really describe that. What I will tell you about is work that Adil has done with surface-borne odor tracking. And he's done that with the help of Manaswini, who was a summer student, who actually was the one who started out the project. Okay, so as you've already seen, there are different strategies that one might adopt. And this is the strategy that you've seen already, where you cast back and forth across the source. If the animal were doing something pretty clever, it might be able to just sniff there, detect a gradient, sniff there, detect a gradient. But to detect a gradient, it would be very advantageous to the animal to be able to do stereo, because then you could see in one sniff that the odor is higher there than there, and thereby correct itself. And of course, if we were doing it perfectly, it would just go straight along an odor trail. So this is what Adil did. He cannibalized an old dot matrix printer, made a treadmill of it. So here's the spool of paper for the printer. There's the camera over there. There's the rat over there. And the brown line is drawn in chocolate. So rats like chocolate. And the experimental design, the way you train the rats, and this is a remarkable thing. There's very little training involved. He draws this line of chocolate and then puts pieces of chocolate at various intervals on the trail. So the rat very, very quickly, without any further shaping and all that stuff, figures out that if it follows the trail, it will get chocolate. Wonderful. Okay, so let me show you what this looks like. Okay, so you can see that the rat is very, very good at doing this. And in fact, the accuracy is on average about 15 millimeters, which is considerably better than humans do, or for that matter, dogs do. Okay. Now, there's a question which everybody should be asking, which is, come on, somebody ask me the question. Huh? Visual input, exactly, yes. White chocolate. No, no, no. That would be an interesting, we didn't think of that one. Okay, so this is infrared. So in fact, there's two possible cues. One is the visual cue, And the other is that chocolate actually leaves a little bit of a texture on the paper. So Adil, in this case, used a liquid odorant, which does not leave any texture, and he used a syringe pump to make sure that the trail was very, very even. So anyway, as you could see, the rat does perfectly well on this. And it can track it. And it turns out that it doesn't matter whether it's doing chocolate or any other odor. Once you've trained it on chocolate, you can transfer the training to something else. And it doesn't care whether it's in the dark or in the light. So it doesn't use any of these things. These are all statistically equivalent. So the accuracy is essentially the same. And so here's the stereo angle. Why don't I just show you the movie, and you can judge for yourselves. So this is the same kind of experiment where we stitch one nostril. Again, in the dark. So the rat's able to do it, no question about that. But I think you can just see and cast your minds back to what it was doing earlier. It just has a larger error. So it's doing the task, it's just making, it's just not as accurate. And it turns out that the deviation almost not quite doubles on average. So here's the thing in the control condition, and this is one nostril shut, and it's zigzagging a bit further back and forth. So good. So stereo is good for real-world tasks. So I would normally, that sort of addresses that part of it. But there's a lot of interesting things one can get from a behavior like this. One question we asked was, to make life difficult, what happens when the rat encounters a break in the trail? So it turns out that the rat starts to scan back and forth even further. And I hope I'm not running out of time, but let me see if I can show you that. So this is done in slow motion. So there's the gap. And you can see that the rat very, very quickly figures out, A, that it's lost the trail, and then B, when it hits the trail again, it immediately latches on. It knows it right away. And we did other nasty things. We gave it bifurcating trails and all these kinds of things. So to cut a very long story short, here are some of the summary points of this, that they track accurately, they use stereo. And what we are starting to infer from this is that rats are not just passively, sampling and then adjusting a little bit. They are actually tracking using some kind of a mental model of where this trail is going. And this is, this you can see to some extent from the fact that a good fraction of cases where there's this symmetrical bifurcation, the animals actually continue down the middle until they accidentally encounter one side or the other. And you saw it also in the gap case where they also continue down the middle. So this leads us to start to ask what I think is one of the most interesting ways of thinking about brain processing, which is the brain isn't just encoding information. The brain is, I believe, as a general way of thinking about it, the brain is actually making models of the external world. The brain, and in this case, the rat is trying to optimize its tracking, not simply by doing very, very rapid sampling, although it's doing that, but by predicting, by doing an intelligent guess about where things are going to go. So we said, let's see how we can analyze this in terms of the actual sampling that the animal is doing, and then try to make a model of it. So we did the usual kind of thing, implant thermocouples, implant recording electrodes, and so on. I won't discuss the recording electrodes, but I will discuss the sampling. So what that tells us is, when does the animal take a sniff? When is it? And from that, since we know where the trail is, and we know where the animal's nose is, and we know when it is sniffing, we can actually get an estimate of what it is detecting, what each sample looks like. And so this is what it looks like. So each of the little black dots indicates a point where it sniffed as it zigzagged back and forth across the trail. So one interesting thing is that it's doing this very close to optimal. So the optimal thing, I shouldn't use the word, we've been beaten up enough about the word optimal. It's doing it very efficiently. One very efficient way to do it would be you sample twice to know whether you're going up the gradient or down the gradient. In other words, are you going closer or further from the odor source? And so that would be 2 samples every time you deviate from the trail. The animal is doing actually For one complete cycle, it's doing of the order of 5. So it's not too far from what is a very, very efficient strategy. Well, that's nice. So what we did then was to actually make a mathematical model, a computer model of what the animal is doing based on what we knew about where it is sampling, how frequently it is sampling, and where the odor trail is. And this model, so this is the model and that's the real data. This model actually works pretty well. It swings back and forth across a trail. It follows bends. And very interestingly, when it encounters a gap, it does something very similar to what the rat does. It starts to swing back and forth until it hits the odor trail again. It does some other things. It made some other interesting predictions. What happens if there are parallel trails? It actually predicts that correctly as well. Okay, so to wrap up, what I think this leads to is sort of where we're going to go with this in the future, is that when we are sampling in a natural context, in a changing environment, we're not just passively sitting there waiting for information to come to us. We actively go out and sample the world. We actively go out and decide what we're going to measure. It's not just letting the information flow in. And we do this in order to update, to improve our simulation, our model of what the world is like. And it's not just encoding data coming in, it's trying to make sense of the world by having an internal model of what is out there. And this is what we're trying to understand the neural basis of. Okay, so I'd like to thank all of you and the people who did the work. Adil, Partha, Manaswini, Urvashi, Raghav, and James, and the people who gave me money. And especially Ubaid, who I'll now end with another little anecdote, which is that when Ubaid was coming around to New York, when I was doing my postdoc to persuade me to come here, so he went out to a little cafe, and as such things happen, we got into a very interesting discussion about science and olfaction and science in general and working in India. And we had such a good discussion that they threw us out after we sat there for more than 2 hours, and they wanted other customers to come in. So, thank you.
Yeah.
Upi, there was a very sharp periodicity in your model trajectory. Is there a similar one in your actual trajectories?
No, there isn't all that much periodicity in the actual data. It depends a lot on how clean the odor trail is. In the model, we just had a line which bent. So I think that you would have more lack of periodicity if you had a more realistic scatter of the odor trail. We tried
predicting a straight trajectory. Supposing you had a sharply curved thing, do you think the rat would learn?
Well, you can't have it go. Partly we're restricted by the fact that the sheet itself is only so wide. So it bends fast. It's a very wide trail, and we made a 5 centimeter wide trail. Long in the middle, it's perfectly happy as the edge.
faculty and PIs on the campus. The canteen is, of course, also open, and the rest of the program will announce as the day goes. It's also up on the website. Thank you. We'll be back here at Apurva. Shall I say 2:20? We're running really late. 2:15. It's just half an hour. Okay. Eat light and eat quick. 2:15.
It'll take some time to.
Because it looks as though there is
Hello. Hello. Apurva. Yeah.
Hello.